Welcome to Gairdner Science Week 2022, the International Symposium. I'm Jim Woodgate. I'm the President and Scientific Director of the Terry Fox Research Institute, and also a Senior Scientist at the Lunenfeld-Tannenbaum Research Institute. So before we begin, I'd like to take a moment for us to acknowledge that, that Mars is located in Toronto on the traditional territory of many nations, including the Mississaugas of Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendell peoples, and it's now home to many diverse First Nations, Inuit, and Métis people. This territory is the subject of the Dish and One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. So this event is part of the Gairdner Science Week, which is an annual week-long celebration of our Gairdner Award laureates, including Dr. John Dick here in the front row. And I'd like to thank our Gairdner Science Week partners, which include the Government of Canada, the Government of Quebec, Government of Alberta, and CIHR IRC. So thank you also to this year's platinum sponsors, which include the University Health Network, RBC Future Launch, Cranbrook Foundation, and the TELUS Health. And gold sponsors, York University, the Terry Fox Research Institute, yay, Queen's University, London Drugs, Roche, CAMH, McMaster University, McGill University, Sick Kids Research Institute, the University of Pennsylvania, University of Toronto, Moderna, and Guardant Health. And finally, I'd like to thank you all for our Science Week sponsors. Without you, really, this important week would be of networking, engagement, and education wouldn't be possible. So today's symposium topic is inspired by the work of Dr. John Dick, who is one of this year's International Award laureates. You will hear from some of the world's top researchers in the field of blood stem cells about the evolution over the years of the concept of the multipotent blood stem cell, first, of course, put forward by Jim Till and McCulloch, and how this has impacted our understanding of blood-borne diseases ever since. So to start things off, I'd like to very much welcome Dr. Ben Ebert, who's Chair of Medical Oncology at the Dana-Farber Cancer Institute, is the George P. Canellis and Jean Canellis Professor of Medicine at Harvard Medical School. He's a Howard Hughes Medical Institute investigator and an institute member of the Broad Institute. And he's going to tell us about clonal hematopoiesis and the origin of blood cancer. Dr. Ebert.
All right, great. Well, I wanted to thank the organizers for the invitation to participate here. It's an enormous honor to get to participate with this group, and wanted to first congratulate John Dick and Stuart Orkin for the Gairdner Award. They are absolute giants in the field, and actually the first talk beautifully combined some of their contributions, not only the stemness, but the identification of von Willebrand factor expressing stem cells that Stu cloned von Willebrand factor when David Ginsburg was a postdoctoral fellow in his lab. But both of them have been extremely meaningful for my career as I followed their work from the earliest stages of my training, but very soon after becoming an independent investigator, I had an opportunity to collaborate with both of them, and the way in which both of them treated me as a colleague and a collaborator when I was just starting and they were already leaders in their fields was enormously meaningful to me. And I think both have taken on incredibly complex fields and established the standards of rigor and experimental approaches that the entire fields have adopted, which is enormously powerful. Both have been mentors to so many current leading investigators around the world. And both have the ability to take incredibly complex experimental biological results and distill biological principles from them that again have become part of our textbooks and how we understand biology, which has been an incredible talent for them both in speaking and writing to articulate those principles. So congratulations to them, incredibly well deserved. These are my disclosures. 1016 Bio is interested in somatic mosaicism. So this is a very basic slide, but important, I think, to how we view the progression of malignancies. If the circles are individual hematopoietic stem cells in cartoon form and time is on the x-axis, you can see that all of our stem cells are constantly acquiring somatic mutations such that every one of our stem cells is barcoded with its own unique set of somatic mutations. Occasionally, those somatic mutations lead to a clonal expansion and an increased number of hematopoietic stem cells with a particular mutation. That does not cause cancer in almost any circumstances, except maybe some rare fusion proteins. But one of the cells within that clone may acquire a second mutation. One of the cells in that clone may require another, and only after the serial acquisition of multiple mutations do we get full-blown malignancies. That has a number of implications, one of which is that we get orders of magnitude more clonal expansions with initiating mutations than we do full-blown hematologic malignancies. And another is that when we do have a full-blown hematologic malignancy, the bone marrow is incredibly complex. There are cells with, say, 5 mutations, cells with 4, cells with 3, 2, 1, and that has profound implications for therapy and recurrence. This field was really opened up a lot by the availability of large-scale sequencing data. This is our first study in collaboration with David Altshuler at the Broad Institute, right as exome sequencing was starting to happen at scale. This was an analysis of 17,000 exomes where we were able to look for the mutations that occur in leukemia or any other hematologic malignancy and look in the peripheral blood by exome sequencing, because exome sequencing for germline predisposition is generally done on peripheral blood cells. And we can see that by the age of 60, about 10% of everybody has a clone in their blood bearing a mutation that we see in acute myeloid leukemia. And these are large clones with, on average, 20% of all the peripheral blood cells bearing the mutation. So these are not tiny clones. So far more common than we expected. This was in about 17,000 exomes, which we thought was a massive study at the time. As a field, we've analyzed well over 700,000 exomes to date. The findings have stayed remarkably robust in terms of the age distribution, the genes that are mutated led by DNMT3A, TET2, and ASXL1, the associations with risk of hematologic malignancy, the associations with overall mortality and other outcomes. So that was exome sequencing. The next stage was that people started looking with deeper sequencing methodologies to see if we can see these clones in the peripheral blood. And the deeper we sequence, the more we find, perhaps not surprisingly. So with whole exome sequencing, we can find clones with a variant allele fraction of 2 to 3%. Whole genome sequencing, generally shallower, a little bit larger clones that we can detect. But if we use error-corrected sequencing and sequence very deeply, clonal hematopoiesis with a somatic mutation that drives leukemia is essentially universal. All of us will have a DNMT3A mutated cell, TET2 mutated cell, probably many of them within our bone marrows. So it is not, certainly when I was learning and thinking about cancer biology at the beginning, I thought it was bad luck that somebody might have the misfortune to get an oncogene or a tumor suppressor mutation in a hematopoietic stem cell that might lead to leukemia. It turns out that's universal. We are all going to mutate DNMT3A many, many times over the course of our lifetime in individual hematopoietic stem cells. What the question is which of those cells is going to expand large enough and which of them will have sequential mutations. So given that everybody has these mutations and they have significant clinical significance, we took a first stab at defining this, and this year the WHO and the International Consensus Conferences define these entities. CHIP is clonal hematopoiesis of indeterminate potential, defines it as not having a hematologic malignancy, because leukemia is also clonal hematopoiesis, but that's not what we're talking about. And we need a lower threshold, otherwise we all have it. And so we initially defined it as having a somatic mutation associated with the hematologic malignancy at a variant allele fraction of at least 2%. These are heterozygous mutations generally, so that's 4% of the blood cells that would have the mutation. In fact, most of the clinical consequences happen with bigger clones, usually with a VAF of greater than 10%, but this is how we first defined it, and it's been adopted. CCUS is the same thing with a cytopenia and is associated with increased risk of malignancy and other outcomes. So that could be the end of the story, but in terms of the types of clonal hematopoiesis, but it turns out there are many types of clonal hematopoiesis. That was just the somatic point mutations in individual genes, but a separate field grew up looking at SNP array data, and there's even more samples that have been analyzed by SNP arrays for groups that are looking at germline predisposition, many, many hundreds of thousands, now into the millions. And approaches have been developed to look for copy number changes that are somatic, not germline, in peripheral blood DNA by SNP arrays, and there are detectable copy number changes in regions that have been recurrently found in hematologic malignancies. It turns out that that is mainly associated with chronic lymphocytic leukemia, CLL, and a little less with myeloid, but it's present in both, and I'll tell you about that. What may be even more common than both of those is expanded clones in our blood that have no known drivers. Those may have drivers that we don't know about yet, and really elegant work from Peter Campbell would suggest that most of them do have some driver that we don't know about. They could be a clone just that's more fit and based on their epigenetic state that's maintained over years and decades, but that is extremely common. So first I'll talk just briefly about myeloid and lymphoid clonal chip or point mutations. The UK Biobank has been just an enormously valuable resource for this field. There's now about 450,000 samples that have been analyzed by exome sequencing and SNP array data with clinical outcomes and well-annotated clinical data. So if we look not only for the myeloid mutations, but specifically for the mutations that occur in lymphoid malignancies, we can detect those in the blood as well. So they're not just myeloid mutations that we can detect. The lymphoid mutations are much less common, and unlike the myeloid ones, there aren't just a few genes that dominate. DNMT3A, TET2, and ASXL1 are incredibly powerful. They're by far the most common mutations in myeloid chip, but in lymphoid, it's a smattering of mutations. Just like in the myeloid side, it's age-associated, as you can see in the panel on the right. If we look at the risk of malignancies, you can see that the myeloid mutations, as you would expect, are powerfully associated with the development of a myeloid malignancy, and lymphoid mutations are associated with the development of lymphoid malignancy. Not surprising, but the associations are very powerful. If we look at copy number abnormalities, this field had called them MCAs, or mosaic copy number abnormalities. The cancer people would just call them copy number abnormalities. But if we look for the myeloid copy number abnormalities that we know occur in myeloid malignancies, like 5q, 7q, 20q, we can find those in the peripheral blood of healthy individuals, and they have a very high risk of developing a myeloid malignancy, up to 30-fold. And on the lymphoid side, we can see that the copy number abnormalities that are associated predominantly with CLL are detectable in the peripheral blood and associated with an enormous risk of developing lymphoid malignancies. If we look for individuals with these abnormalities plus an abnormal lymphocyte count, the risk of CLL goes up to about 1,000-fold. The concern in this particular data set is that some of those individuals may be undiagnosed CLL. And so we're now working with colleagues who have large-scale cohorts with flow cytometry data to understand for sure which of those individuals have no premalignant state, which have MBL, monoclonal B-cell lymphocytosis, and which have CLL, and whether these genetic findings can refine what we consider a premalignant state for CLL. So one of the unexpected but really exciting aspect of this field has been the association of CHIP and as well as myeloid malignancies, it turns out, with inflammatory disorders. And perhaps one of the most surprising aspects of this is that the majority of the morbidity and mortality associated with somatic mutations that lead to leukemia may be due to non-malignant consequences. So this was apparent in our very first study, where we saw that having one of these mutations in the blood, having CHIP, was associated with increased overall mortality, and quite striking, about a 40% increased risk of dying of all causes, and that stayed robust through many data sets. That's a bigger shift in overall mortality than you can explain by leukemia. We all study leukemia, and it's an incredibly important and challenging disease, but it is not that common to shift overall mortality. But what we did find was that there was an increased risk of cardiovascular disease, both coronary artery disease and ischemic stroke, with a substantial hazard ratio correcting for all the known risk factors. This was not the goal of the study, and sort of subject to all the statistical criticisms of searching through the data for an explanation for the association on overall mortality. But we then validated this in 4 additional cohorts. These are prospective cohorts, just like the UK Biobank, so blood drawn at the beginning of a study that was analyzed genetically, and patients were followed prospectively to see whether they had, in this case, heart attacks or not. And each of the 4 cohorts replicated the finding that having CHIP at the beginning of this study increased the subsequent risk of coronary artery disease and having a myocardial infarction. I'd say that now that there's 8 or 10 cohorts that have been analyzed, the associations obviously will vary across the cohorts. The sicker they are, the older and greater risk factors, CHIP will contribute more. Young, healthy people, there's less of an effect, though still apparent. And the other critical factor in these studies is how well clinically annotated these studies are. These particular studies were developed for cardiovascular disease, so they're very well annotated for outcomes. If we rely only on ICD-9 or ICD-10 codes, the data get, not surprisingly, a little bit messier. So the next question is, if there's this unexpected association between CHIP and cardiovascular disease, maybe this is just aging, maybe there isn't a causative association. So we and others took an approach of trying to validate this in mouse models. Mice don't get cardiovascular disease at eating their normal chow and their healthy lives in our vivariums. But if you knock out the LDL receptor, feed them what people call a high-fat diet for mice, or a Western diet for mice, they do develop robust atherosclerosis within a reasonable time frame. And when we transplant in either wild type or TET2 knockout bone marrow, we get very different results. So TET2 knockout blood cells transplanted into LDL receptor knockout mice get very robust atherosclerosis. Heterozygous knockout of TET2 does the same, and wild type much less so. And this has now been replicated by many labs using slightly different experimental paradigms in terms of the diet. DNMT3A does the same, though interestingly, heterozygous DNMT3A mice do not do this, homozygous do. ASXL1, JAK2 have the same phenotype, and so this is at least as a model system, has become very robust. So is this limited to cardiovascular disease? I'll note another experimental approach that we and others have taken. So one approach has been to take large cohorts of individuals with exome or other types of sequencing data, find the individuals with CHIP, and look at various outcomes. Another way is to use epidemiology data, and this is a study that we did using Medicare SEER data. And in this case, we know the individuals in the United States who have MDS, and we can look using Medicare data at whether they had any new diagnoses of inflammatory diseases that occurred in the 5 years prior to MDS, the time frame we would think that they might have CHIP before they got their myeloid malignancy. And inflammaging is a way that people have combined many of these disorders led by expression of inflammatory cytokines like interleukin-1 beta. So when we did this, looking at the new diagnosis 5 years antecedent to the diagnosis of a myeloid malignancy, a number of inflammatory diseases have higher prevalence before their diagnosis of either MDS or CMML, including cardiovascular disease, pulmonary diseases, some metabolic diseases. Interestingly, neurodegenerative diseases go the other direction. There is a decreased risk of Alzheimer's and other neurodegenerative diseases in individuals who go on to develop a myeloid malignancy. And that is completely independent work from Sid Jaiswal's lab, was presented as a plenary at ASH last year, found the same thing in cohorts that were actually developed for studying Alzheimer's disease. And there was a striking decreased risk of Alzheimer's disease in individuals with CHIP. And there are very high VAFs of these mutations in the brain. So these mutated cells are in the brain at a high frequency. They're doing something that seems to actually decrease the risk of Alzheimer's and other neurodegenerative disease. If you focus in, you can see that ischemic heart failure, as well as atherosclerosis, emphysema, kidney disease, fatty liver disease, osteoarthritis, osteoporosis, rheumatoid arthritis, and gout all had significant associations. This has been true in both CHIP studies and prospective cohorts, as well as these epidemiology studies. The effect sizes are modest, 30 or 40% increased risk, but in aggregate, given that CHIP is very common, this is a very large source of morbidity and mortality. We and others have developed a large number of mouse models of some of these disorders. We can see it with DNMT3A, which has got a stronger association with osteoporosis. We can transplant mice with DNMT3A knockout, bone marrow, and by micro-CT actually see increased osteoporosis. And TET2 transplanted mice who are exposed to cigarette smoke, we can see an exaggerated COPD with actual airspace disease. And so this has become quite robust that particularly with TET2, many inflammatory phenotypes are exaggerated. So what could be mediating that? Well, one of the very recurrent findings has been that if you look at cytokine levels or gene expression or single-cell RNA-seq, interleukin-1 beta and other downstream consequences of the NLRP3 inflammasome have increased levels in the blood or in the bone marrow of TET2 knockout cells. This is true in bone marrow-derived macrophages, in vivo, or in the bone marrow. So interleukin beta seems to be particularly important, as well as a number of other chemokines and cytokines that have actually already been associated with atherosclerosis and other inflammatory disorders. Here's another human genetic study from Alex Bick and Pradeep Natarajan. This is the top med cohort, another 100,000 whole genomes in this case. Again, same genes are mutated, same age association. But a subset of these individuals had serum cytokines measured. And I just point out in particular, can't see my own cursor, this red dot here, which is the IL-1 beta levels in TET2 mutant in individuals with TET2 mutant CHIP. So particularly elevated interleukin-1 beta levels in individuals with TET2 mutant CHIP, very much correlating with our mouse models where interleukin-1 beta is particularly elevated in the TET2 knockout mouse models. And this was borne out amazingly in a clinical trial of canakinumab. Canakinumab is an IL-1 beta blocking antibody developed by Novartis, actually in clinical practice from Novartis. And they did a 10,000 person randomized controlled clinical trial of canakinumab to decrease cardiovascular events. Very expensive, very large trial. It met its endpoint, barely, of decreasing cardiovascular events, major cardiovascular events. The first trial of an anti-inflammatory showing a benefit in cardiovascular disease. Well, when our paper came out, they went back and exome sequenced the samples from their study, and they found that individuals without CHIP in the upper left did not benefit from canakinumab. But the ones with individuals with TET2 mutations had a greater than 60% decreased risk of major cardiovascular events and really drove the effect. And in fact, individuals with CHIP without a TET2 mutation didn't have much benefit. So TET2 is special. It seems to elaborate higher levels of interleukin-1 beta. The Walsh lab has shown that inhibition of the NLRP3 inflammasome, which is upstream of IL-1 beta release, will also block the TET2-mediated increased atherosclerosis in the TET2 mutant model. So at least in the TET2 mutant cases, there seems to be a particular role for interleukin-1 beta, and blockade of either interleukin-1 beta or the inflammasome may have benefit. And then finally, wanted to quickly review some of the factors that have very reproducibly been associated with poor outcome. One of which, for hematologists, is somewhat satisfying, is RDW, the red cell distribution width. So mean cell volume, MCV, really shifts if most of the cells are changed. But the red cell distribution width shows the variation in red blood cell size. That has been associated with cardiovascular disease and increased mortality for many decades in epidemiology studies. But what we find is that CHIP is associated with an elevated RDW, maybe because 10 or 20% of the cells are mutated and they're making larger and smaller red blood cells. But it's the individuals with an elevated RDW and CHIP that have the really high risk. And that's convenient because we measure RDW with every blood count routinely in all of our patients. So a very easily obtained parameter. So we think that combining those will be incredibly useful clinically. Another finding, this was from Sid Jaiswal's group that I think it was just published, that looked at the methylation clock. So there have been a number of groups that have looked at methylation clock and blood cells. This is just combining all blood cells. And the highest risk is really in individuals with both CHIP and the altered methylation signature. Interestingly, many of these mutations lead to altered methylation directly. Whether that's mediating the effect or not isn't clear. But if you have the aging methylation clock or old methylation signature of an older individual without CHIP, the risk isn't that much greater. But if you have the methylation signature and CHIP, those are the people who are at greatest risk. So probably the RDW in this are accessing some similar biology. And then the mutations are not all equal. This is highlighting some work from Liron Schluch, who trained with John Dick. But we and others have seen very robustly different mutations associated with different risks. So TET2, DNMT3A are associated with an increased risk of getting leukemia, but it's relatively modest. Other genes, including splicing factor mutations and P53, are associated with really dramatically increased risk. And now that we can start to subset, if you look at RDW, the identity of the mutations, whether there's 2 mutations, the size of the clone, we can identify a small subset of individuals who are almost guaranteed have a 70–80% chance of going on to getting a myeloid malignancy. Whereas others, for example, individuals with a single DNMT3A mutation and nothing else, have almost no risk. Very, very minor increased risk. So I think we're going to make a lot of progress in the coming years at stratifying these individuals and understanding what diseases particular individuals are at risk for, and hopefully how we can intervene to decrease risk in specific individuals. So I wanted to note that CHIP is extremely common and very age-associated, associated with an increased risk of hematologic malignancy, overall mortality, cardiovascular disease, COPD and other inflammatory disorders, and with aberrant expression of inflammatory cytokines, including interleukin-1 beta. The CHIP mutations and mosaic copy number abnormalities independently increase the risk of hematologic malignancies, that the myeloid mutations or copy number abnormalities associated with myeloid malignancies, the lymphoid ones, are predominantly associated with CLL, not other lymphoid malignancies, which is not surprising because CLL is a circulating tumor, nodal lymphomas probably have less circulating cells and therefore less easily detected in the peripheral blood. The risk of progression to malignancy and other bad outcomes is associated with particular features, including specific genetic alterations such as splicing factor mutations in p53. Multiple genetic abnormalities are associated with increased risk. The size of the clone is associated with increased risk, and altered CBC perimeters, in particular, the RDW. And wanted to thank everybody in my lab. Sid Jaiswal led this work when he was a postdoc in my lab and continues to be a leader in the field in his own lab at Stanford. We work closely with Pradeep Natarajan at the Broad Institute and MGH, with Alex Bick at Vanderbilt and Peter Libby at the Brigham. I'm happy to take any questions.
Time for one very quick question. That's fascinating.
Oh, that one.
So, whereas the minor chip mutations, almost all of them, I think, can be tracked back to stem cells. I assume lymphoid chips mutations are not typically found in cells.
That is a great question, and one that we were. Oh, so the question was, myeloid mutations can be tracked back to stem cells. They are present in multiple lineages. They are present over many, many decades of life, and in some cases have been tracked from childhood through old age. So meets a number of criteria for stemness. The lymphoid mutations have not been analyzed properly, but we are actively doing that, and that's a great question. So the lymphoid mutations are associated not with cardiovascular disease, but increased risk of infection. And that's been shown by a number of groups now. CLL is also associated with increased risk of infection. You could say that is a lymphoid thing. That is aberrant T and B cell function, and that explains increased risk of infection of everything, sepsis, COVID, broad infection risk. Alternatively, those initiating mutations could happen in a stem cell, and that's been hypothesized for many years. Maybe the later mutations occur in a committed B cell, but that initiating mutation maybe could happen in a stem cell, and then conceivably could have myeloid abnormalities as well as lymphoid. But that remains to be determined, but it's a good question. But it also raises an interesting question of cell of origin. It's not one thing. The initiating mutation may happen in one cell type, and the progression mutation may have in a separate cell type. Thanks.
Thank you to Dr. Ebert. So next we have Professor Anna Tsvee, who's a professor at the University of Copenhagen and a group leader at the Biotechnology Research and Innovation Center. And she's going to discuss investigation of human fetal blood development at the single cell level.
This is not really, okay. First, I would like to thank the organizers for inviting me here today. And yeah, so in my group, we are interested in how blood cells develop and how is their development and ultimately their function influenced or determined by environment and environmental cues. And to address these questions, we are using single-cell multi-omics approaches. So initially, single-cell RNA-seq, which allows us to look at the transcriptome on individual cells, but later on, single-cell ATAC-seq, when we can look at the chromatin accessibility, whole genome sequencing, which allows us to look at the somatic mutations and use them to perform lineage tracing experiments in human, and most recently, single-cell spatial transcriptomics, or spatial transcriptomics with the single-cell resolution. And application of these technologies really allows us to move from this rigid view of hematopoiesis, which was actually introduced very nicely at the beginning of this session, and then start to look more at the process of cell phagocytosis and differentiation as a gradual process where stem cells and progenitor cells transit gradually from one cell state to another before they emerge as a fully mature cells. And in the lab, we are looking these processes in two very different contexts. One is in solid cancers, where we are looking at the behavior and changes or reprogramming of myeloid population and their interaction with the tumor cells, and the other is human fetal blood development, which is actually a very interesting model to look at the blood production, because it's well established that stem cells during fetal development have much better and higher self-renewal potential compared to the adult stem cells, and in that sense they're of great value for regenerative medicine. Now, just to briefly explain, the first stem cells in human appear around 27 days post-conception, and this is happening in dorsal aorta. This is the place where stem cells are going to be specified, as well as some progenitors, but they're not going to expand in this area. To expand, they need to move into the fetal liver, and this is happening very soon after their specification, already at 4 weeks post-conception. And these stem cells and progenitor cells here are going to further expand in their numbers, but they're also going to differentiate to allow the growth and growth of the embryo. And then around 10.5 weeks post-conception, again, these stem cells and progenitor cells are going to move into the bone marrow, which is after birth going to become only site of hematopoiesis, obviously in health. But what is interesting is that between 10.5 weeks post-conception up until birth, both liver and bone marrow will be active sites of hematopoiesis, and that really allows us to look at the behavior of stem cells in two very different environments, but having them actually coming from the same fetus, meaning at the same stage of development. And so in a way, in the two studies that I'm going to describe, we look at the transcriptional and epigenetic changes which are happening in stem cells and progenitors as they're migrating from one environment to another, as well as somatic mutations that they are acquiring, which allowed us to actually reconstruct the phylogenetic tree of human fetal blood development. So in the first study, we collected stem cells and progenitor cells. Actually, we used over 23 different phenotypically well-defined populations to isolate individual cells from fetal liver and bone marrow from 10 fetuses from 17 to 22 weeks post-conception. And what you're looking at here is inferred differentiation trajectory, where each dot here represents individual cell, and obviously each color represents a different cluster, which is then annotated, so therefore it's a different cell type. And what we were able to do here is just using kind of typical methods in computational biology when we are analyzing single-cell transcriptional data is to infer this differentiation trajectory and then start looking at the changes in the gene expression of some of the marker genes that are identified in each of these populations across pseudotimes. So pseudotime, as it was already well introduced in the first session, is allowing us to look at the differentiation trajectory by looking at the changes in a transcriptome of cells as they are migrating from one cell state to another. And what that was nicely actually showing is that we were able to identify three main paths which are going from the stem cell population. We call them here MEMP, so these are megakaryocyte erythroid mast cell progenitor population, granulocyte progenitors, and then lymphoid and myeloid progenitor populations. And what was interesting about these three progenitor populations is that they were oligopotent and highly proliferative. Now, when we look at the expression of the marker genes, what we were able to see is that, as you're seeing here, pseudotime, is that there was a very nice high expression of typical hematopoietic stem cell markers in stem cells, and they're being downregulated. The cells are differentiating, and then there is upregulation of important lineage-specific markers. Now, what was interesting here for us, and you can see that from this graph, but also after we did subclustering of our stem cell population, is that actually within these hematopoietic stem cells from fetuses, we could not see different subpopulations of stem cells. So we could not see on a transcriptional level something that is called priming or bias of the stem cells. And so to us, it looked actually that on a transcriptional level, this was a homogeneous population of cells. The second important thing was that when we actually connected our phenotypic markers with the transcriptional signatures that we were seeing, it turned out actually that phenotypic stem cells were very heterogeneous population of cells, and this was true for any of the progenitor populations that we looked at, suggesting that these markers which are used to isolate stem cells in adult are not maybe the best markers to actually define stem cells during fetal development. And this really kind of suggested that we need a different maybe cell set of markers to define stem cells during fetal development. And so to do that, we went back to our single-cell RNA-seq data, to our so to say, transcriptionally homogeneous population of stem cells. And we looked at the markers which are specifically expressed in this population and defined a new panel for stem cells, which we call here CDREF. And then we did another experiment in which we collected fetuses and stem cells from fetal liver and bone marrow based on our new panel of CD antibodies. And then we, on one side, took half of the population and did single-cell RNA sequencing on them, and did, with the other half, single-cell in vitro differentiation assays. And what our single-cell in vitro differentiation assays showed was that around 90% of cells formed colonies, and many of these colonies were multilineage, and actually the output was independent on the feeder layer that we were using, whether that was MS5 or human fetal mesenchymal stem cells, or actually if there is no feeder layer, the output was same in the first 2 weeks. But also when we sequenced our now stem cells from the new panel that we defined, and we mapped that back onto our UMAP that I was showing you earlier, we could see actually the majority of these cells, over 90%, were falling back in the stem cell compartment, suggesting that indeed we now have a more pure population, on a transcriptional level at least, of stem cells. And when we now had this new panel, we were able to collect new fetuses and now isolate stem cells using this new panel from liver and from bone marrow, and to look at the proliferation properties of these cells. And what we were able to show is that the cells from liver compared to those from the femur, even though they were collected from the same fetus, meaning at the same stage of development, were actually proliferating more in liver compared to the femur, suggesting that this migration is already early in development, causing the slowing down of the cycling of the stem cells. Now, I was saying that there was no heterogeneity in our population of stem cells on a transcriptional level, but we wanted to see if there are any changes on the kind of epigenetic or the level of the chromatin accessibility. And to do that, we did single-cell ATAC-seq, where we now collected individual cells, again, from the liver and from the femur, from fetuses. In this case, we were focusing on CD34 positive, CD38 negative cells with the idea to enrich for the stem cells and progenitor populations. And so, as in the single-cell RNA sequencing, each cell here, each dot here represents a single cell, but here, instead of looking at the differentially expressed genes in our cluster, we are looking at the differential accessibility of the peaks. Now, following the clustering, what was really challenging when one is working with the single-cell ATAC-seq data and chromatin accessibility is to annotate cell types, to know which cell type actually each of these clusters belongs to. So we wanted then to use the same rationale that we use when we were annotating single-cell RNA-seq data. So here, what you can see is the same list of genes that I was showing in the initial plot when we were annotating single-cell RNA-seq data. And what was very obvious is that the markers, which are very good to define stem cells on a transcriptional level, are not actually that useful in defining stem cells and different progenitor cells when we look at the chromatin accessibility. And the main reason was that when we look across these 7 clusters, we could see that all of these genes were either open or they were closed. So there were not enough resolution here to actually look at the different populations. So we decided to have a different approach. We decided to actually use our single-cell RNA-seq data to inform the identity of the cells from the single-cell ATAC-seq data. And to do that, we used the so-called anchoring method, which allowed us to integrate single-cell RNA-seq and single-cell ATAC-seq data, where you're putting these 2 data sets in the same space and then perform a label transfer of from the cells from the single-cell RNA-seq to single-cell ATAC-seq. And what that actually showed us is that the percentage of different population was as expected, so that was good. But what was interesting is that when we looked at actually the distribution of different cell types across these 7 clusters, we could see that actually our stem cells were present across all 7 clusters, suggesting that within this population of transcriptionally homogeneous stem cells, there was actually different subpopulations which were based on the chromatin accessibility. And actually, to be more specific, the differences were coming from the transcription factor activity when we were looking at the openness of the transcription factor motif sequences. So what we could see is that in these 7 clusters, there were differences in the transcription factor activity of different transcription factors which are relevant for different lineages, suggesting that actually the heterogeneity is coming maybe from another layer of information from the epigenome rather than from the transcriptome. And so to kind of look at this more closely, we decided to actually focus on GATA1 regulans. So GATA1 is a very important transcription factor for the megakaryocytic, erythroid, and mast cell lineage. And we wanted to compare what is happening between the expression of the GATA1 regulans. So these are the genes which are regulated by GATA1 in terms of the chromatin accessibility and their gene expression. And so what we could see is then the stem cells, when we move from stem cells to megakaryocyte erythroid mast cell progenitors to megakaryocytes, for example, if you follow this path, we could see that many of the genes which are regulated by GATA1 are not expressed in stem cells, but are highly expressed in megakaryocytes. So this is fine, this is what we expect. But what we were able actually also to see is that in our cluster 1, which we predict to be our stem cells in our chromatin accessibility map, is that already in stem cells we could see that these promoter regions for these genes were already open, suggesting that actually, although there was no transcription of these genes, these cells in a way were kind of primed to, in kind of future steps, express these genes. And so some of the key points here is that by using single-cell RNA-seq and ATAC-seq, we were able to generate epigenetic and a transcriptional map for human fetal liver and bone marrow hematopoiesis. And we could show that the stem cells were differentiating into 3 different oligopotent, highly proliferative populations. And interestingly, that the changes actually in the motif accessibility in stem cells are preceding the changes which are happening on a transcriptional level. And in that sense, maybe suggests that the priming is happening at this level. And finally, using single-cell RNA-seq data and single-cell in vitro differentiation assays, we were able to refine the strategy for sorting stem cells with around 90% purity at the transcriptional level. I should point out that this study was done between 17 to 22 weeks post-conception fetuses, and the studies that we are now actually doing suggesting that this panel of the markers that one can use for the stem cells is actually changing during development. And this one is most probably kind of limited to the window that we were looking at. Now I'm going to kind of do a little bit of a change in the narrative, and I'm going to talk about a different study in which we actually wanted to look at this migration of the stem cells from one environment to another one, but using a different approach, using somatic mutations as a way to do the lineage tracing of these cells during development. And basically, the whole concept of the lineage tracing is based on a very simple idea, and that is that if a stem cell or a progenitor cell acquires mutation, that mutation is going to be inherited by all the progeny of that cell, and then if any of the progeny of that cell acquires additional mutation, then their progeny is going to inherit that mutation. And so basically what that means is if anyone takes blood from at any stage in our life, whether that's a fetal development or later on in adulthood, what we can use is that if we have the information about all the mutations that are present in these stem cells or progenitor cells, then we can reconstruct a phylogenetic tree, which will actually allow us to go to the very beginning. And the very beginning in the context of the human development is actually zygote. And so what we are able to then do is to look at the first division of the zygote to blastocysts, and in a way to reconstruct actually human fetal embryonic and fetal development. And so just what you're seeing here is a very simplified version of the human embryonic and fetal development. And there are just a couple of points that I would like to make here. So basically what is known from previous studies is that around 100 cell stage, there is a separation, physical separation between trophoblast and inner cell mass. And this is a very important point in development because trophoblast is going to give rise to a fetal part of the placenta and is going to give rise to chorionic sac, whereas the inner cell mass is the one that is going to give the embryo. And so this is important because that means that any mutations that are acquired before trophoblast and inner cell mass are separated, they will be present in all the tissues that originated from these two different populations of cells. Whereas, for example, mutations that have been acquired only after trophoblast separated from the inner cell mass, they will be only present in the fetal part of the placenta and in chorionic sac. And so furthermore, then inner cell mass will separate into the hypoblast and epiblast, and epiblast will give rise to germ layers, ectoderm, mesoderm, and endoderm. And then, for example, blood is of mesodermal origin. Whereas for the hypoblast, when it comes to human fetal development, there is a little bit of controversy where it is known that the hypoblast is giving extraembryonic mesoderm, but there were speculations that they're also giving rise to primitive blood, which actually in mouse is originating from the epiblast. And using these somatic mutations, we think that we actually managed to shed some light on this and propose that actually primitive blood, as you will see later on, originates in human from the hypoblast. So to perform this experiment, we had to design it in a very particular way. So we collected one fetus which is 8 and another one which is 18 weeks post-conception. And we collected hematopoietic organs, liver and bone marrow. But from the same tissue, from the same fetus, we also collected tissues which have a different embryonic origin, in a way covering this whole map that I was showing you earlier. And then from the hematopoietic organs, we collected individual cells, we grew them into colonies, and then we did the whole genome sequencing, so we know which mutations are present in these blood colonies. But for the tissues, we fixed these tissues, embedded them, sectioned them, and did the laser capture microdissection, which allowed us to have a very precise information about the embryonic origin of each of these tissues. And then we tested which of the mutations that are present in the blood are also present in these various tissues. And based on the somatic mutations from the colonies, we reconstructed phylogenetic tree. But to further interpret our results, we really needed this information from other tissues. So how did this all look like at the end? So these are two phylogenetic trees, one from 8 and one from 18 weeks post-conception. And before I move into some of the main findings, just to give a brief kind of explanation of what we are looking at here. So these at the end are terminal branches. So these are the colonies that we sequenced. The length of these branches is proportional to the number of mutations that we are detecting. And we are starting with the notion that the beginning of, or the top of this phylogenetic tree are two blastomeres. So these are the first two cells that originated after division of the zygote. And so these are our kind of two cells. And so the first thing that was obvious from just looking at these two trees is that there is a different contribution of these two blastomeres in these two different fetuses to the blood compartment. Whereas in this fetus, one blastomere gave rise to 1/3 of the blood compartment and the other to the 2/3. Here, one blastomere contributed to 99.9% of all blood cells, whereas the other one had really small contribution. And so the first conclusion that we were able to make here is that really the contribution of the blastomeres to the blood compartment and to the embryo of whole is very plastic and can be very different. The second thing that we were noticing is that there was a difference in the rate of mutation acquisition across development. In other words, what we were able to see is that the rate of mutation acquisition was much higher in the first 3 cell divisions, so around here, compared to the following period. And initially in the first 3 divisions, this is around 2.4 mutations per cell division, whereas later on it drops to 0.9 mutations. And you can then see here in this area here, we have these polytomies, which in this context could be defined as divisions in which no mutations have been acquired. Now, the third thing that we could actually see here is what is the number of mutations that are acquired in stem cells during development. And the average number for the fetuses at 8 weeks post-conception is around 24 mutations, whereas that number is obviously higher in 18 weeks, and it's around 42 mutations per stem cell. Now, what we couldn't say from these phylogenetic trees is when were these mutations acquired. And this is where the information from the mutation sharing between stem cells and other tissues became actually very relevant. So just going back to my previous slide, really the idea here is that if the tissues are sharing fewer mutation with cells which descended from the ancestral line of descent of the blood, which we call here blood antecedents. So if they share fewer mutations, that means that they separated from the blood earlier on. So for example, we would expect that the tissues that originated from the trophoblast share fewer mutations with stem cells than, for example, tissues which came from epiblast, any of these other tissues. So basically to present this visually, what we were able to do is now to look which of the mutations are shared across various tissues. And as an example here, for example, these mutations in red are all the mutations that are shared between tissues which originated from the trophoblast and our blood colonies, which means that these mutations were acquired before trophoblast and inner cell mass separated. And basically, by using this logic, we can actually time the divergence of embryonic and extra-embryonic lineages during development. And so this is where additional information actually came from in terms of the origin of the primitive blood in human, which we were also able to collect from the heart of one of the fetuses. And so a different way to present the figure that I was showing you earlier is this way, where we can look at the cell generation. So this is a cell division. So the first cell generation is the first cell division, so 2 cells. 4 cells, 8, 16 cells. And here we can see what fraction of the cells in various tissues share mutations with our blood. And so what we could see is that the tissues that originate from the trophoblast, 90% of trophoblast lineages were lost by the 4th generation. And this is 16 cell stage. And this is very important because if you remember from the beginning of this part of the talk, I was saying that the physical separation of trophoblast and inner cell mass is happening around 100 cell stage. And we are actually seeing that this cell phase choice is happening much earlier. This number is around 10% for post-gastrulation tissue, so only 10% of post-gastrulation tissue is lost from the tree by the 4th generation. But when we look at the placental villi mesenchyme, which are originating from the hypoblast, and when we look at the peripheral blood, we can see that they're sitting in between epiblast and trophoblast. And this is actually where we hypothesize that actually this primitive blood is originating from the hypoblast, the same as these placental villi mesenchyme. So I'm going to just finish here with some main conclusions from this part of the talk. So we know now that tens of somatic mutations are accumulated in the first 8 weeks of human development. We know that there is unequal contribution of each blastomere to the blood compartment, but actually to the embryo as a whole. We know that the mutation rate is not constant during development. It's much higher in the first 3 cell division and then drops down. We could actually estimate, I was not showing this data, the number of blood antecedents at different stages of embryonic development. And our results, in a way, suggest or are in line with the idea that the primitive wave of human hematopoiesis actually originates from the hypoblast and not from the epiblast, like as it's the case in mouse. And so I would like to thank some of my current and previous lab members. And the first part of the talk was done in collaboration with Judith from Heidelberg, and the second one with Peter Campbell from Sanger Institute. Thank you for your attention, and happy to take your questions.
Time for a question. From John.
Very nice, Anna. I was just wondering, in the first part of your talk where you had identified this new set of markers capture fetal stem cells, you ran out clonal and in vitro assays. I wonder whether you have done any sort of in vivo functionality to self-renewal and those more traditional assays.
No, we didn't. But it's something to do, yeah.
Thank you, Professor Serge. So next we have Dr. Emmanuelle Passegué. She's the endowed alumni professor of genetics and development and director of the Columbia Stem Cell Initiative at Columbia University, Irving Medical Center in New York City. And she's going to tell us about deregulated hematopoietic stem cell behavior in aging and malignancies.
Thank you. And thanks to the organizer, it's my absolute delight to be here for this meeting which celebrates John's achievement and work. John has been very inspirational for my career from my early day as a postdoctoral fellow in Weissman Lab. So I've always looked forward to his publication and his fantastic work. So I want to tell you today a bit in continuation of what has already been approached by Stein Erik, which is this changing view of hematopoietic development from this hierarchy with stem cell at the top and going through this very strict commitment through progenitor population to produce all the different type of blood cell, which kind of hide a lot of cellular biology and change in activity of the cell when they go through this differentiation process. And here you can see running in the movie the enormous granulocytic output, which can come from like one hematopoietic stem cell seeded on a stroma layer. And now the single cell revolution that Anna mentioned, which really put transcriptional signature on this developmental path and basically is challenging the concept of very defined step of differentiation and this biphasic change in lineage behavior when you get to this fate commitment event. So what we have worked a lot is on this notion that you really have a lot of change happening when you get to a regenerative context. So most people focus on steady state hematopoiesis and the structure of the system. But in fact, there is humongous change when you challenge the system to really amplify one lineage component. And in the case I want to talk to you is really the granulocytic lineage, talking about emergency myelopoiesis. So each stage of the hierarchy has a very much defined function. The stem cell compartment, which is the transplantable units that we need in the clinic for hematopoietic regeneration, is very much a metabolic switch. Basically, the engrafting stem cell reside in this quiescence phase of the cell cycle, and they mostly run on a glycolytic program for their source of energy, with some as well help of fatty acid oxidation. But mostly it's glycolytic. But to activate, to produce a downstream blood progenitor, they really need to change a lot of their biology. They have metabolic remodeling, they switch to an oxidative phosphorylation driven metabolism, a change in the associated with cell cycle activation, a change in their epigenetic poisoning, and all these features together lead to a loss of self-renewal, a loss of transplanting activity. In contrast with, again, is the ability to differentiate, produce a downstream lineage-committed multipotent progenitor, which have already been mentioned. In the mouse, there are this alphabet soup of MPPs, 2, 3, 4, and so forth. They can be isolated with very defined surface marker, leading to these different populations recognizable on FACS plot. But basically, that's the stage where lineage, like generalization, lineage commitment to various blood output is taking place. And finally, in the oligopotent progenitor compartment, you have this phenomenon of lineage amplification. And the one that we have the most interested is the granulocyte macrophage progenitor population, which on a flow plot appear as a fairly homogeneous population, but it's not. And in fact, its biology in vivo is very very much illustrative, that you have a lot of cell biology, expansion in some GMP patch where you have localized foci in the marrow where you have self-renewing GMP, which really amplify in number without differentiating, and then undergo this GMP cluster formation, which is when they stop expanding, they block the self-renewing program, and they symmetrically differentiate through this like differentiation and produce an outburst of granulocyte. So, of course, this doesn't happen in a vacuum. There is a lot of influence, the bone marrow niche microenvironment, which have not been talked about a lot so far, which is really an ecology of cell type, which will play essential role in controlling the activity of the stem and progenitor cell. The cytokine milieu, Ben beautifully illustrates the role of inflammation in driving the clonal selection in the context of clonal hematopoiesis. A lot of these factors directly talk to stem and early multipotent progenitor to really educate their activity, and of course the molecular wiring of this emergency myelopoiesis pathway. So, how is that characteristic? The hallmark filter of this activation process, you have, as I say, stem cell engaged activation with metabolic remodeling of the compartment, overproduction of the more myeloid-biased multipotent progenitor, MPP2, MPP3, the myeloid reprogramming of the lymphoid-biased multipotent progenitor, MPP4, which is switch away from his ability to produce lymphoid cell toward producing myeloid cell, and our favorite, this GMP cluster formation, which is really the big output of like granulocyte production. A lot of regulatory mechanisms, I say, and this is a constantly growing list with activation signal, which are mostly cytokine, SCF, IL-1, TNF-alpha, all of that act at the level of the stem cell to activate them, to force them to engage into this regenerative program. There is modulatory factor, interferons, for example, or Notch-Wnt signaling ratio, or the self-renewing network, IRF, beta-catenin, at the level of the GMP cluster. So at various stages of this hierarchy, we are mapping the mechanism which controls that. And most as important are the repressive factors, which are basically returning the system to homeostasis. And that's a very important aspect. You activate, you overproduce your myeloid cell, but then you need to stop this process to revert to the steady state hematopoiesis. And in this context, megakaryocytes are extremely important, and megakaryocytic base factor like platelet factor 4 or TGF-beta are essential to put back stem cell into the quiescence phase and terminate this process during normal regeneration. So, as I say, we follow that by imaging in the marrow and the formation of GMP cluster, and you can see that in every context of a regenerative response. Stem cell transplantation in lethally irradiated recipient, 2-4 weeks post-transplantation, you see this mix of GMP patch forming GMP cluster, leading to the myeloid outburst, which is characteristic to this early phase of the regenerative response following transplantation. 5-FU-mediated myeloablation, a chemotherapeutic compound, you give one shot, it damages the normal marrow, a lot of change now in the niche, a lot of secretion of factor, but you start to see in the, on the top of this damaged niche, and in fact, on very dysmorphic megakaryocytes, that which we maybe don't see very well in this image, but are underneath of this GMP patch, this purple aggregate of cell, which then very quickly start to form this cluster where the first differentiating granulocytes are around the border, forming like this vast GMP cluster, and finally the granulocytic outburst. And finally, you can do the same just by triggering myeloid cell production with GCSF stimulation, one injection, where you see again this very nicely forming GMP patch, which drives the granulocytic outburst. This process is observed in every myeloid malignant context that we have looked at. Here I'm giving you an example of our favorite inducible bicerebral model, which model human chronic myelogenous leukemia, which is a DOX-inducible model. We see this remodeling of the hierarchy. The pre-leukemic stem cell is at the level of the hematopoietic stem cell. They are decreasing in number in the bone marrow because the bone marrow as well develop myelofibrosis, so reduced number of stem cells can survive in this challenging niche, but the one which are left really activate emergency myelopoiesis pathway with overproduction of myeloid-biased MPP3, not really change in the production of the lymphoid-biased MPP4 in numbers, but very much change in their myeloidization, in the sense that in colony-forming activity, they form much more myeloid colony, and downstream of this early stem and progenitor cell, this massive formation of GMP clusters that we see in the bone of this animal. We see similar, albeit slightly different because of the block of differentiation in the context of an acute myeloid leukemia. This is the most utilized model in the field, which is this MLL IF9 transduction transplantation model, which is a one-hit event driving fully malignant, fully transformed AML. So we see an erasure of the early stage of the hematopoietic hierarchy with persistence, as very nicely described by Scott Armstrong, of a leukemic stem cell population at the previous level of a granulocyte macrophage progenitor population. And this leukemic stem cell population fuels this like amorphous GMP cluster formation, where we have this dysmorphic GMP patch poorly turning into GMP cluster in the marrow of this transplanted AML recipient mice. So we can model AML development from the standpoint of this hijacking of emergency myelopoiesis pathway with preleukemic mutation, cell cycle activation, epigenetic poisoning, leading to this enhanced clonal dominance at the level of the stem cell compartment, activation of the myeloid bias multipotent progenitor, formation of GMP cluster, which are acquiring additional mutation, leading to this leukemic stem cell emergence, and this blast production, and this collapse of the hematopoietic hierarchy in AML development. So another way of showing that is very much this evolution from this acute activation of emergency myelopoiesis, which is really a feature of regenerative hematopoiesis, into this stepwise development of malignant disease and the constant engagement of emergency myelopoiesis driving the preleukemic stem cell emergence, and finally at the level of the AML with this dysregulated emergency myelopoiesis feature really influencing clonal competition and emergence of leukemic stem cell from a later stage of hematopoietic differentiation. So another context where this principle are apply is physiological aging, which we model in the mice, which is really a crosstalk between an inflame and remodel whole bone marrow niche, where here is our single cell analysis of the niche of a young mice, where you have a lot of cell population and very nice ecology and hierarchy, which is very much affected in an old animal, where you see disappearance at the endosteum of a lot of the mesenchymal stroma and osteoprogenitor, which are characteristic of a young healthy niche. And this dysregulated endosteal population really contribute to secretion of a lot of pro-inflammatory factor, this inflammaging signatures that Ben mentioned, a lot of interleukin-1, a lot of TNF-alpha, and so forth. And this inflammatory mediator, especially interleukin-1, contribute in turning the central marrow leptin receptor MSC into a very inflammatory subset, which are very different supportive activity and certainly educative property for the hematopoietic hierarchy. In fact, what we see is in response to this inflamed niche, activation of this emergency myelopoiesis program from an old hematopoietic stem cell compartment, driving the production of myeloid cell at the expense of lymphoid commitment, and I think contributing as well with what Sten-Erik describes, this platelet bias bypass mechanism in changing really the composition of the old blood system. So here we can show you that we have this expanded stem cell population, increasing production of MPP3, decreasing production of MPP4, and constitutive presence of this GMP patch in the bone marrow of an old mice. So we think that this change in the whole microenvironment and the change in the activation status of the whole blood system is really the ground for like the selective pressure leading to the emergence of clonally mutated stem cell in the context of clonal hematopoiesis as described by Ben. So what we are interested is really, can we target this maladaptive activation of emergency myelopoiesis process during malignancy or here in aging? We have spent a lot of time in my lab to really see if changing systemic factor in the blood could have an impact on the deregulated old hematopoietic stem cell function. And we recently published all these results where we use approach such as parabiosis, plasma transfer, or intervention, systemic interventions like caloric restriction and exercise, which are very popular in the aging field to really reawaken some other stem cell population or change physiological aging output. And in the case of the blood system, we really saw absolutely no effect of this intervention on neither the old hematopoietic stem cell regenerative potential as measured by transplantation or the whole blood composition, and this unbalance in terms of lineage output. What we found instead is that blocking niche-mediated inflammation, and in this case interleukin-1 signaling, has a very pronounced effect and can really rescue whole blood production. So we did either short-term acute inhibition with anakinra, which is I think a preceding version of anakinra that Ben mentioned. So only 2 weeks treatment, but in the context of a regenerative challenge, such as a 5FU challenge, this lead in the old mice, which is really bad at responding to 5FU, really lead to bone marrow attrition. We see a much better regenerative response, much more recovery of cellularity, lymphoid cell production, red blood cell production, so a very much ameliorate blood regenerative response, blocking interleukin-1 signaling. And when we look in old interleukin-1 receptor deficient mice, we have a much more attenuated aging niche feature as well, indicating that blocking inflammation will limit the aging of the niche, the inflammation resulting from the aging of the niche, and therefore the dysregulation or imbalance of lineage production resulting from this niche effect on the whole blood system. So, but to go further in this kind of understanding and targeting maladaptive emergency myelofibrosis, we really need to understand better the regulatory mechanism controlling the output of the system. And this brings me to the last story I want to tell you, which is what we have learned when we took a closer view at the MPP3 compartment. So as I show you with the first plot in the previous slide, we have nice cell surface marker to isolate a population. But this population is highly variable, and our first insight that they were subset with different activity was when we did transmission electron microscopy, and we uncover like a third of this population and having this very striking morphology full of ear, endoplasmic reticulum, which are nicely lined up with ribosome, indicating a lot of protein synthesis and a very strong suggestion of a secretory morphology. Basically, at a much smaller cell, at a much smaller scale, this looks like a plasma cell, which is a factory for immunoglobulin production. These are not plasma cells. These do not produce immunoglobulin, but they have the morphology of the secretory cell. So this brought us back to a work published a few years back by the lab of David Baltimore, which shows that you could take LSK in this case, so the top like HSPC stem and progenitor cell, stimulate them with a strong inflammatory challenge, LPS and PAMC3, and get secretion from this early stem and progenitor cell. So we refined his approach by separating all the subpopulation we were interested. Globally, in terms of secretion, without an inflammatory challenge, is extremely low. When you put an inflammatory challenge, you get a lot of secretion from GMP, which is expected. The myeloid progenitor is the first intermediate along the myeloid hierarchy that secrete. But in the HSPC compartment, MPP3 stimulated with this challenge is the most secretory component subset of this hierarchy. So these are what the MPP3 secrete. A lot of them are cytokines which promote myeloid differentiation. They have as well repressed some cytokine secretion. They are all associated with a lymphoid commitment. We know because we use ER tracker to separate secretory from non-secretory subset in the MPP3 that the most secretory subset is this ERI, this R endoplasmic reticulum compartment subset. And here is two factors that we mostly use to track the secretion of this population, our favorite, TNF-alpha and interleukin 6. So we use a co-culture experiment with harvested tissue supernatant from cultured MPP3 and MPP4 that to seed naive cell in methylcellulose or in liquid culture. And what we could see is that in response to MPP3 secretome, basically stem cell, MPP3, MPP4, naive cell are all pushed to undergo myeloid differentiation. And we can see that in methylcellulose and in liquid culture as well, where MPP3 supernatant from stimulated cell, not from unstimulated cell, really push cell to undergo myeloid differentiation. We as well develop an imaging approach which allows us to recognize the various MPP subset on thin section. And what we can see is that they are located in the vicinity of stem and other multipotent progenitor. And this result match with what is coming out from the lab of Dan Lucas, which much better imaging resolution, which shows that like stem and multipotent progenitor are long-range cells. They don't aggregate, they don't form cluster. They really migrate away from each other. On average, these cells, these MPP3, are 150 micron away from stem cell, and that's really where they are in the marrow. And we think that the secretory subset of MPP3 is really there to educate the local neighborhood by secreting this cytokine to like force them to undergo myeloid differentiation. This like heterogeneity in function is matched by a molecular heterogeneity by single-cell RNA-seq analysis, where we see that 2/3 of the MPP3 population has this immature feature, and then we have 1/3 which follows this intermediate, metabolically activated intermediate 2 myeloid prime subset. We identify a cell surface marker, which is Fc gamma receptor, which really track this secretory MPP3. When we look when they are in this PC plot by bulk RNA-seq, this ER low, which are this immature cell, are much closer from a stem cell behavior versus this ERI, which are this Fc gamma receptor positive, are much closer to the GMP feature, while when we take the bulk MPP3 unseparated, we have this mixed identity, which is away from all of three. And in fact, when we project an MPP3 on a UMAP of unfractionated HSPC, they map everywhere, so they don't appear as a distinct population because of this heterogeneity we describe here. But when we do this kind of like mapping into etoamide MPP single cell data set, we can see that the ER low immature cell kind of like connect to all of the other myeloid progenitor versus the ERI really are close to the GMP. But they are not GMP. We have a lot of data demonstrating that they are very distinct from GMP. They are upstream. They are basically a bypass mechanism from a true multipotent MPP3, which is capable of giving rise to CMP, MEP, and to go through steady set differentiation versus this Fc gamma receptor positive ERI MPP3 is really this bypass mechanism which acts through lineage priming and secretion of cytokine, which reinforce myeloid cell commitment. Of course, this is as well found in myeloid malignancy. Went back to our pre-leukemic CML model, this inducible BCR-able model, where I already show you we have increased MPP3 production. All this increase is really due to this expansion of this Fc gamma receptor secretory MPP3. This cell secrete constitutively some cytokine, which are not found in a wild-type non-leukemic MPP3. And these cytokines that they constitutively secrete push naive cell to undergo myeloid differentiation, but nowhere to the same extent that cells stimulated by the strong inflammatory challenge. So, but it's a much more restricted subset of cytokine. Within all of them, we were particularly interested for many reasons by galactin-3, which is one of these constitutively secreted cytokine by a leukemic MPP3. And when we remove galactin-3 from our leukemic model, we significantly extend the survival of the mice, very similar to what we already observed when we remove IL-6 from the system of these leukemic mice. And most importantly, we really turn off the GMP cluster formation process in this leukemic animal, where it's very prominent in the normal BCR-able mice, but it's pretty much gone when we remove this leukemic MPP3. Sorry, secreted cytokine. So this is a model we build about this myeloid bypass, which amplify myeloid differentiation in inflammatory and leukemic condition through similar, although slightly different mechanisms in terms of like the molecular underpinning of the process, but very important. And the last thing I want to leave you is really with the central role of this emergency myelopoiesis pathway activation as a convergence point between niche deregulation, inflammation, and likely driver of clonal competition and aberrant differentiation in myeloid malignancy and aging. We have still a lot of work to do to really turn this knowledge, to weaponize this knowledge in intervention for patient benefit. And that's why I want to show my lab, which I help a lot with this process, and some empty chair in our lab retreat on Fido Island for new postdoctoral fellow. I understood a lot of students are attending this meeting, so I make a pitch there. Really thanks my lab. Yuna Khan did all the work on the secretory MPP3. She is now a new assistant professor at WashU, and she's continuing on a lot of this work, and a lot of thanks to the rest of the team for their help. Thank you for your attention.
We've got time for maybe one question before the break. Okay. So I'm sure Dr. Passegué will be happy to answer questions during the break. So that's going to conclude the first part of today's symposium. I'd like to thank all of our wonderful speakers, incredibly engaging, lots of really new data. And you've given us a lot to think about over the break. So we're going to take 10 minutes. So we'll be back at 11:05. And the next session will start then. Thank you.
Okay, everyone, if I could get you to cease your networking, and we'll try to get ourselves moving to the next part of the amazing symposium. It's a remarkable thing for me to be surrounded by this amazing amount of terrific science, the leading edge of what's happening around the world. So my name is John Dick. I'm a senior scientist, Canada Research Chair at Princess Margaret Cancer Centre, and university professor at the University of Toronto and professor in the Department of Molecular Genetics. One of the recipients of the 2022 Gairdner Award. I have to say what the Gairdner people are telling me here. Okay, now I'm really embarrassed. But I am thrilled to chair the second session of this symposium, which is a topic which is just near and dear, obviously, to my heart. As I said, to be introducing my good friends. And one of those is the next speaker, Dr. David Scadden from MGH. He is the Gerald and Darlene Jordan Professor of Medicine at Harvard University, Professor and Chair Emeritus of the Department of Stem Cell and Regenerative Biology, was the co-founder and co-director of the Harvard Stem Cell Institute, hematologist, oncologist, MGH, directs the Center for Regenerative Medicine, and an amazing scientist and good friend. We've known each other for a long time. David.
Well, thank you so much, John. And I'm truly honored to be a part of this symposium today, and particularly delighted to have a chance to do this to honor John and his amazing contributions in STU, and they are two of my personal heroes, I have to say. I mean, they've been the people who have done the most innovative, the most rigorous work, and have also been really outstanding mentors for junior scientists. So these are two people without whom the world would be very different in terms of our thinking about the blood, and frankly, our thinking about the development of malignancies. So spectacular to be a part of this. So I'm a physician scientist by background, and I've been very interested in the environments that provide cues for hematopoietic stem cells, and in particular have focused in the past on bone marrow. But we've turned our attention more recently into thinking about a very different kind of microenvironment, and one that is very peculiar, and that is of the thymus, where we know this unusual organ is one that really has a very different aging process than most of our other organs, in that it has a decline that begins actually in our childhood, and at the advent of puberty goes through a very dramatic change in which there is an atrophy of the cell population, the shrinking in the size of the thymus, and really replacement of epithelial cells that make up the cortex and medullary regions by adipose tissue. So that unusual early senescence of or involution and destruction of the tissue is something that results in very dramatic changes in terms of our ability to produce the critically important subpopulation of cells, alpha beta T cells. And we know that in all of us, as we decline, as we have a change in age, that in addition to accumulating the somatic injuries that we've heard about from Ben, there's also a collapsing of the T cell repertoire. So if you look at the sequencing of the TCRs, they will decline inexorably with time. And that, of course, leads to then a more patchy, if you will, ability to respond to different challenges. That's certainly been evident in age with our ability to respond to vaccines, COVID, increased incidence of autoimmunity, and, of course, the reactivation of viruses, and potentially also the increased incidence of cancer. But patients who suffer particularly from this are those who get an insult to their immune system, and that occurs acutely in the setting of stem cell transplant. So there we know that individuals who have essentially their existing T cell population wiped out during transplant conditioning have a difficult time recovering T cells from their donor, and that actually puts them at high risk for a number of different problems in terms of both graft versus host disease and infection. So we've been interested in T cell neogenesis, and the interesting element of this is that generating these types of cells depends on two distinct organs, one being the bone marrow, where a precursor population that is T cell competent is generated in the bone marrow. It then exits the bone marrow, circulates in the blood, finds its way, and must seed a new organ, the thymus, where it undergoes the serial steps of differentiation of mature T cells that then enter the blood. Now, this idea of there being a two-component element to this is something we found intriguing, particularly because most emphasis to try to regenerate the thymus has been focused on the thymus itself, and particularly its epithelial components. But we were really inspired by work that was done here at the University of Toronto by Juan Carlos Zuniga-Fluker, who has done some outstanding work of being able to generate T-competent precursors ex vivo. And what he has demonstrated is that in a very atrophic thymus here characterized by its medullary and cortical regions defined by different staining with cytokeratins, which becomes very dysmorphic in a atrophic thymus, that if you simply take the precursor populations that are normally coming from the bone marrow and that are in the blood, and he makes them ex vivo, injects them into the blood, what happens is the crosstalk between the hematopoietic cell and its epithelial tissue is that it reorganizes this tissue. There is a restoration of the balance and organization grossly of the thymus, and a regeneration of new T cells. So there is an ability to essentially foster the regeneration of the thymus by hematopoietic cells. So we then decided to look at whether or not we could define ways of enhancing essentially the supply side of cells getting into the thymus with two different approaches. One is to look at whether or not we could enhance the number of cells that are generated by the bone marrow or a bone marrow equivalent, and then also look at ways in which we could enhance the seeding process through identifying a niche within the thymus that recruits hematopoietic cells. So I'll tell you these two vignettes. So the first is really work that was based on efforts by my lab and others to identify what populations of the hematopoietic system in the bone marrow, the non-hematopoietic parts of the bone marrow that contribute to the generation of T-competent cells. And through a number of different manipulations, Vioni Yu was able to knock out particular subsets of cells that are stromal cells and look at the impact of that take-one-out strategy on the generation of lymphoid cells. And she found that if you knock out these osteocalcin-positive cells, which are osteoblastic in nature, that there was a decrease in B cell production, but also a decrease in T cell production. And she found if she knocked out DLL4, this Notch ligand, specifically on those osteocalcin-positive cells, she ended up having a reduction in the number of these T-competent progenitors, as well as reduced expression of the cell's surface receptors that are an important part of the trafficking process. So with that information in hand, we work closely then with Dave Mooney, who's a spectacular chemical engineer who works on generating biodirective materials. We had a shared postdoc, Nisarg Shah, who then decided what we would try to do would be to create something that was material, but enhance the generation of a T-competent bone marrow organoid. And so this was basically to try to create such a material and then see whether it has an impact on thymic production of new T cells. And so the way this is done, Dave is an expert in the use of alginate, which is a complex polysaccharide derived from brown seaweed. You can then cross-link it with various different chemical cross-linkers. If you then put it in presence of water and do a controlled freeze, the ice crystals that are formed by the water create then a macroporous structure, and it's very elastic depending on the degree of which you have the cross-linking of the alginate. So we could do that and create then what is essentially a sponge with the alginate creating the superstructure of that sponge. And you can then put into the sponge something like BMP2, a growth factor that's known to both recruit cells, so mesenchymal cells could be recruited into the area, and as well as it enhances angiogenesis, and it then induces those cells, instructs them to become bone. In addition, you could have attached to it a particular ligand. In this case, we conjugated to the alginate the Notch ligand DLL4. So with that, if you now have this material, it's actually so collapsible that you can put it into a syringe. And when you put it into a syringe and inject it, it will reassume its shape. And so that allowed us to be able to now take this material with these proteins, put it under the skin of an animal, inject it, and it reassumes this pillow-like shape. As we do that, we can then look over time, and fairly rapidly, this turns into bone. So if you do a micro-CT, you see this very smooth surface, egg-like structure. It has, on higher magnification, the pink here being the bone, and then there's trabecular areas that you can see here. And if you look at even higher magnification, you see that what is within this is actually hematopoietic cells. So this is indistinguishable from normal bone marrow in terms of the different cellular elements. And if you now test this in a setting of transplant, where you could lethally irradiate an animal, give it a bone marrow from a donor that has GFP positivity, and then do the subcutaneous injection of the cryogel at the same time you do the transplant, you can now look to see whether or not cells engraft and enter the cryogel, so-called BMC, bone marrow cryogel. And they do. And so you can find here these GFP positive cells. They're only found, though, if bone forms, if the BMP2 is present. Now, these are mostly myeloid cells. And if we look for the cells we're most interested in, the common lymphoid cells, these are only found if you have DLL4 present. And with that, you see a marked elevation in the abundance of these cells by greater than 100-fold. So what's the impact of that? We could demonstrate that these cells do migrate from this little organoid under the skin to the thymus, and then they ultimately spill out into the blood. And they do so in a way that leads to a recovery of circulating T cells at a rate that's much more rapid than if you give a transplant alone, or do the empty gel or the gel with just the BMP2. So you can increase the number of T cells. The question is whether you're increasing existing cells or making new cells. We can measure with a simple biometric tool called the TREC analysis. So when the T cell receptor rearranges, it cleaves out a portion of the endogenous locus, and that portion actually religates to itself, forming a so-called T cell receptor excision circle, or TREC. Now that structure does not replicate when the cell replicates. So it's a very good quantitative tool to measure T cell neogenesis. And it's actually now used clinically as well to see if people are making new T cells. And when we looked for those in our in the thymus of our animals. This is an unmanipulated animal, totally normal animal. If you do the transplant alone, it's clearly not making many cells. But if you have the cryogel with the BMP2 or the DL04, it's indistinguishable really from the unmanipulated animal. You could also address the question of whether or not you're making a bigger T cell receptor repertoire. And you can do that by looking at these areas of recombination, the joint regions between these. And you can then, by sequencing, quantitate the number of different new TCRs that are present. And doing this on this type of three-dimensional plot where you can see the different bars obviously represent different subspecies of VJ rearrangements, and the height is the amount of them. This is an unmanipulated animal. This is an animal that's gone through transplant, and these are a very barren landscape. It changes in terms of the height of some of these if you have the BMP2 alone, but not much in complexity. But if you have the two present to combine, you end up with a complexity that is very reminiscent, at least, of what exists in the unmanipulated animal. So that was very encouraging. And the idea then that maybe this is providing new T cell competence was something we also then measured by vaccinating the animals with a new antigen, in this case, ovalbumin, where you can quantitate the number of T cells that recognize the very specific epitope on ovalbumin. And you can do that by flow cytometry. And an unmanipulated animal clearly has a much more robust response. But a transplanted animal does not have any response. And we could see with ours that you do get at least something more substantial. So it did seem you're providing new T cell competence with this cryogel. If we compare it to the process of making progenitors ex vivo with this system developed by Juan Carlos, and then injecting those cells in the blood, we think we also do better than that. So this is important, but if you're increasing reactivity and you want to use this in a setting of transplant, of course you might be concerned that you would in fact exacerbate something like graft versus host disease. So we tested that in a model of a major MHC mismatch, allogeneic transplant in the mouse. And what we found quite unexpectedly is that you actually blunt graft versus host disease and you markedly improve survival in the animals that have the cryogel. And at least trying to investigate why that might be, at least it could be attributed in part to the fact that we are not just getting T helper and cytotoxic T cells, but also T regulatory cells to a much greater abundance than with the transplant alone. So we think this was interesting in terms of our ability to balance production of cells that are capable of reactivity to new challenges, as well as dampening response to challenges that can be excessive and pathogenic. Seeing if this works in human cells, we took advantage of making xenogenetic models pioneered by John and Tsvee, and looked at whether or not this cryogel can enhance T cell generation with the xenogenetic transplant of human stem cells. It does seem to do that. You get a CD4-CD8 ratio. Ours, most normal people, is around 2, but in transplant it's often lower. And in these models it's often 1, so with the cryogel we could increase that. We could increase the production of T regulatory cells, and we could see again an impact on what is usually a roaring graft versus host disease in this xenotransplant model. So we think this then is a way in which you can actually use information about a defined bone marrow niche to create a material. This is not cells, it's a material that you can use that's biodirective, and it creates something of an organoid then that has this ability to improve T cell generation in the repertoire and makes a difference in terms of post-transplant setting. The advantage of this is that it is cell-free. There's no matching with the donor or recipient needed, and there's no cell culture. In fact, we've frozen it down, FedExed it to ourselves. You can then use it again, and it works fine. So it's quite stable, and it can be administered subcutaneously. You can also remove it. It's not tethered into the deep muscle, for example. And we think it may be useful, something of a means of delivering an augmentation of bone marrow function to enhance the generation of these cells that then go on to foster restoration of the thymic function. So that's one strategy that we've worked on. Again, that's a materials approach. The other, we wanted to define whether or not we could modify or understand some of the processes that lead to cells that leave the bone marrow and enter the circulation to find their way to the thymus. A lot's been done about the receptors and ligands that are needed for that process. We wanted to understand if we could understand the cells that contribute to that and define a thymic niche. So this is work done by Karen Gustafson in the lab. And what she has done is really focused on mesenchymal cells because in development, it was shown by Laureate decades ago, that the mesenchymal cells are critical in terms of making a thymus. That if you remove these neurocrest-derived mesenchymal cells, the third pharyngeal pouch, which is the basis for the generation of the thymus and parathyroid, does not result in the changes of the endoderm here necessary to create the primordium of those two organs. And so you end up having you do not form a thymus. So they're essential in development. And the question is whether they also play an essential role in an adult thymic niche. So she, to look at this, did single-cell analysis of human and mouse thymus. And she did this in a way that demonstrated that mesenchymal cells, which are in purple on both of these, are actually a very important, large component of the non-hematopoietic parts of the thymus. This is better quantitated here in the pie charts, again, of human and mouse. And then this is a single-cell analysis. But by flow cytometry, you can also see that this is the majority population, the thymic mesenchymal cells. Much smaller are the thymic epithelial cells that we tend to focus on. So she then asked, are there subsets within these? And yes, if you do single-cell analysis, there are three groups. There's one, the T48 positive cell, that really seems to be the fibroblastic superstructure of the thymus. There are these two CD48 negative populations marked by different indicators. This is more of the adipogenic cell population. This cell population, the periostin positive population, has some suggestion, we thought, of having a role potentially in T cell generation. And so we looked at that in terms of expression of different T cell related molecules. And in particular, things like CCL19, which we know are critical for recruitment of cells to the thymus, as well as growth factors, FLT3 ligand, and IL-15, important for T cell proliferation and differentiation. And those are particularly abundant in the periostin positive population within the mesenchymal cells, and are also, at least in the human, actually the majority of the CCL19 is made by these cells compared to other cell types in the thymus. So we then wanted to see if these were a niche cell, we'd have to define it both in terms of their anatomic location as well as their function. And just looking at where these cells reside, this is the gross morphology of the thymus. You can see the medullary cavity is easy to discern and marked particularly by cytokeratin 5. When we look for where our cells reside, the periostin positive populations are clearly abundant in the medulla, but they're also present in the cortex. And they're particularly abundant in lining vessels at the cortical medullary junction, which we know is the place that cells are coming from the blood into the thymus. So these were in an anatomic location consistent with a niche cell. And to define function, we then looked at whether or not they change in abundance in settings where we know the function is impaired. This is the population in magenta that we're going to look at. Under age conditions, these cells tend to go away, which we can see here. This is percentages, so this necessarily increases, but this actually also decreases in absolute number. And the same was true if you look at the animal post-transplant conditioning. Again, magenta, the cells tend to go away, and these periostin positive cells are the ones that are most affected. So then looking at if we could now experimentally delete these cells, we made an animal where we could deplete them based on diphtheria toxin. You depleted these cells, and you ended up getting a change in the ability of cells to move into the thymus, so reduction in the cells that are entering. These are so-called early thymic progenitors, early thymocyte progenitors, and then the next downstream step in making a T cell, the DN1 populations also diminished. So she then wanted to see, well, if these cells are important and if you remove them, they have a negative impact, what about adoptive transfer of them? Does that improve production of T cells, entry of cells into the thymus in a transplant setting? And strikingly, she actually noted that she could use these cell populations and see an increase in these cells that enter the thymus, hematopoietic cells coming from the blood. And this was positive. This was the case whether you used the pink or the periostin positive cells. These are actually more simply identified by CD248 negative, so I'll refer to those. We'll use those in subsequent experiments. But this was certainly above that of other sham injection or sham cell injection. So when we do that, we look at what happens to the circulating cells in the blood in a transplant setting. And again, you can improve the output of both of T cells that are helper cells or CTLs with the presence of these adoptively transferred mesenchymal cells. We could look at whether or not this results in new T cell generation using that method I spoke about before, the quantitative measurement of TRECs. And there again, you see an increase in the number of these TREC positive cells. And you can then also look at the repertoire of the T cell receptor rearrangements that are present. This is a different way of representing the data, but you can see by the complexity of the colors and the size of the different forms that there is a very different complexity of the TCR complexity in the thymus of an unmanipulated animal, very barren under control conditions, and this was somewhat benefited by the presence of these adoptively transferred cells. And this is just the entropy index to demonstrate increased diversity of those. So then does this have an improvement in immune response in these animals? Again, just doing the same ovalbumin vaccine strategy. We could measure that the cells that specifically react to this new antigen were identified as being increased, and their ability to respond to the antigen with production of gamma interferon also increased. Now, one thing I didn't mention, but when we delete CCL19 from these cells, we actually do get a blunting of the effect. CCL19 seems to be really necessary for their function. And what we did is also then take other mesenchymal cells and overexpress CCL19 in them. And in this case, we did this with just a bone marrow cell, since that's a very readily available mesenchymal cell. If we overexpress CCL19 and now just inject it into an aged animal, a mouse with an atrophic thymus from age, no transplant, and look at whether or not we change the number of cells that enter the thymus. This is just looking at a single time point. It's increased. It's nowhere near as vigorous as in a young animal, but it does happen. And actually, if you look at sequential times, it increases more gradually than in a young animal, but it does increase. And interestingly, consistent with the data that Juan Carlos had shown, you get a change not just in the hematopoietic cells, but you also get an improvement in the epithelial cells in the thymus. And you do improve the overall vaccine response in these older animals. So we think that this population of cells does actually represent a niche cell in the thymus that's important for the seeding process of cells coming out of the bone marrow, being able to get into the thymus. We think that that can be something that can enhance the recovery of T cells in a transplant setting. Also, early indications suggest that it may improve the function of an atrophic thymus. And so this idea of being able to improve, if you will, the supply side of cells that get into the thymus may be a way of trying to augment T cell production, which is something relevant, obviously, for the transplant setting, but also for many more settings, and we're in the process of testing some of those. So I want to close by just saying that we've tried these two different approaches. Both are niche-based, one to actually create something of a niche organoid, the other to enhance the niche through adoptive transfer of cells. I want to thank the people who did all the heavy lifting. These are people in my lab. I think I've mentioned those who really led the work. And in particular, our close collaborator, Dave Mooney, has just been fantastic. Peter Karchenko helps us with computational biology, and Charles with imaging, our funders, and then the institutions I'm lucky enough to be associated with. I want to also thank my wonderful lab. I thank you, and I want to specifically say again, congratulations to John and to Stu for your fantastic careers. So thank you.
We have time for some questions. Let me ask the first one. So in the first part where you were transplanting the hydrogels, it looks like you're getting activity quite relatively quickly after you implant them. Do you have a sense of what's the cell that's actually getting in and doing the job?
Yeah. Well, I think so. If we make gels that only make bone, and they don't have the generation of a cavity. They don't actually have any marrow, obviously, and they don't make any new cells. So I think it's macrophages getting in to actually create the cavity that then leads to the generation of the marrow, somewhat similar to the way I think bone forms in development.
Right. But in terms of the T cell side of things, right? I mean,
Well, I think there are stem cells present. And so I think it's actually the DLO4 encourages the specification of those to become T competent. But stem cells are definitely present. And in fact, you can modify what's on the gel to enhance the number of stem cells there or to enhance the number of myeloid cells, depending on what you're interested in.
Go ahead. Ben, go ahead.
Oh, Ben.
I had a related question. Is there something on the surface of the gel that prevents a fibrotic response so that you don't end up with just fibrosis?
Yeah, you do get a little bit of fibrosis around it, but it's not, you don't get an inflammatory response of any substance. And that, there have been reports of using alginate and getting inflammatory responses. It has to do with the purity of the material, it turns out. And so that has not been a big problem. The fibrosis happens, but it doesn't constrain the vascular flow, from what we've been able to tell.
Laszlo, and then.
Hi, Laszlo Emmanuelle, ICR. Oh, fantastic talk. I guess I had two quick questions. Have you tried both the Thigh-MC and the sponge?
Not yet.
And what happens?
Yeah, no, we'd love to do that. We haven't yet had a chance to do it.
Okay.
We're still trying to get the second story published. So.
Yeah. So the other question I had, I guess, are some of the risks because, for example, the sponge with your DLL and BMP, you're now creating or recreating enhanced T cell genesis in an aged thymus microenvironment. Have you looked at whether there's proper negative selection, any issues with enhanced production of autoimmune clones, et cetera, et cetera?
We have not, and we'd love to do that. We think that what we're starting to do, and we have no data on this, is to try to use it in like an EAE model and see whether we exacerbate that or mitigate it, and we just don't know yet.
And Keith, last question.
If you fast forward in your imagination, how do you see this with scale in a human set?
Yeah, right, exactly. Nobody's going to walk around with a new femur. Yeah, right. So we did do the allometric scaling, as for what that's worth, it's modeling. And it turns out that it's about the size of what we use for transplant patients in their ports. It's about the same size, or like an old pacemaker. So that's what we have estimated, our best estimate. That's why we feel we absolutely need to do non-human primates, is to get a sense of whether that will really scale. But I think that would actually be acceptable, right? Because people are used to, or at least we know, people can tolerate walking around with something like that for long periods of time, certainly with the pacemaker and also with the ports. So we'll have to see.
Great. Thank you so much.
Thank you.
Great talk. Last but not least, it is my distinct pleasure to introduce Professor Tsvee Lapidot. He is currently the professor of stem cell biology and regenerative medicine at the Weizmann Institute. You know, the reason that I stand on any of these stages over these last many years, getting recognition and so forth, is directly related and correlated with Tsvee and the seminal contributions that he made to my lab very early on. Tsvee is my second postdoc, and we'll talk a little bit more about that later on today. But it's a distinct pleasure to have him here talking about light and dark. Tsvee.
Thank you, John, for your kind words. And I'm delighted to be back in Toronto. I feel like a pilgrim whenever we land and I see the CN Tower. I get emotional. In our house, we use only Canadian maple syrup. And I have many friends here over the years, but I'd like to start first by congratulating John and Stu for their seminal work, fantastic achievements, and we'll all celebrate that. And in honor of John, I changed my talk a little bit. The first part, I'll talk about the light and darkness, but in the second part, I'll talk about sorted results and the test of time. So what you see here in the first slide, you see two types of asparagus, a green one and a pale yellowish one. And if you look carefully, you see that the pale yellowish one are shorter than the green ones. And they look completely different, but genetically they're identical. And the only difference is the pale yellowish one are grown in the dark and do not receive daylight. And that's why they're less woody and I think also a little bit more expensive. Now, moving from the asparagus, what happens in mice? And what you see in this slide is different times of the day, and already before us, the group of Simon Mendes-Ferrer and the late Paul Frenette have shown that release of stem cells to the circulation, or as we call them, egress, happens only during the day, doesn't happen at night. And we looked at the different time points and we compared daylight to darkness in competitive repopulation assays, and you can see that the night cells repopulate much better than the day cells, which got our interest. What's the reason? And then we looked at metabolism, and again, in the footsteps of Simon and the late Paul Frenette, they have shown that 1 hour after light initiation, there is a wave of norepinephrine in the bone marrow, which we could repeat. And we found that this norepinephrine also leads to a transient wave of TNF, which is a very important cytokine. And as you know, there's elegant work done by Steneric and by Emmanuelle about TNF and hematopoietic stem cells. And this leads to a transient increase in reactive oxygen species that goes down at 11. And surprisingly, we found that darkness onset does something similar. Because you can say there are light receptors, there's something we see with the eyes, but there are no receptors for darkness. Still, we see norepinephrine, TNF, although at lower levels. And if you look at 11 p.m., we get the first clue why the night stem cells are better than the day stem cells. The ROS levels are significantly reduced. The next thing we looked at is the levels of stem cells by cell surface markers, because in a 24-hour period, there's no proliferation. And you can see that the night stem cells have much higher levels of these stem-specific cell markers compared to the day stem cells. Now, the group of Maestroni has shown that the darkness hormone melatonin is also produced in the mouse bone marrow and also in the human bone marrow. And so we measured the levels of melatonin. And of course, this is a night hormone that appears only at night. And you can see huge elevation in the levels. Now, mice are nocturnal. They're active at night. But melatonin doesn't put them to sleep because they don't have the receptors for sleep to melatonin. They only have receptors for other functions of melatonin. So we said, let's see what happens if we give melatonin for 5 hours, just at the onset of light, and then we look at the levels of the stem cells. Again, there's no cell cycle here, but you see there's an increase by surface markers, reduced levels of c-KIT, increased levels of CD150, et cetera. But stem cells are identified in functional transplantation assays. Let's do the transplantation. Here are the control, and here are the repopulation levels from cells from the donor treated with melatonin. You see a significant increase. Even when we take half the cell dose from the marrow of the melatonin-treated mouse, it's similar to the control, and in some cases, even slightly better. Now, this is all mouse, and we wanted to know what happens in humans. I called the headquarters in Toronto, and John and Stephanie repeated our experiment and found the same thing. This is in chimeric immune-deficient mice, the most primitive hematopoietic stem cells in the mouse marrow. They received the melatonin only for 4 hours because Stephanie was on her own and she had a lot of mice. But still, she got a significant effect. You look at the short term, the effect is less significant or not significant statistically. And then you look at progenitors, you have no effect. So to summarize this part of my talk, we look at the bone marrow niche, that it acts as a prism and dynamically regulates, on the one hand, the daily production of immature and maturing blood cells that all have a finite lifespan. And that's what happens during the day in mice. Not only for the stem cells that are released at 11, but for all the myeloid and lymphoid precursors and red cells, etc. While at night, there is replenishment of the reservoir. We need to prepare undifferentiated stem and progenitor cells for tomorrow, and to have this thing ongoing. And there's also a third role for the niche. And again, here, David had pioneering and very important results, is what is the role of the niche, not only from regulating the stem cell activity, but also, and this is something I will talk a little bit about more later, protecting them from DNA-damaging agents and DNA-damaging insults, including radio and chemotherapy, which are very important clinically. Now, we spoke about hematopoietic stem cells and light and darkness. What about the stroma cells in the microenvironment? So while our animal colony light initiates at 6 o'clock in the morning and we have to wake up early, here in Boston, in this pioneer work from 1966, light initiates at 9 o'clock. I guess maybe because of traffic jams or something. And they found 1 hour after light initiation, peak osteoclast frequency. So as you know, there's bone turnover that takes place during day and night, and our bones are destroyed and regrowth, and there's also bone growth. It takes place during the night. So we were pleased to see that peak osteoclasts are also 1 hour after light initiation, where we have this transient inflammatory responses. And there's also a dark side to light and darkness. And this is elegant work done by Ben Ebert, showing that transcription factors for clock genes, BML1, they are essential for a model of AML in mice. And manipulation with these clock genes and transcription factors, the leukemic cells, the leukemic clone is destroyed. So I think here there's a clinical relevance, and I think we'll hear more in the future about this type of manipulation in order to cure patients, which is very important. Now, I will move to host immunity, and this is something now in the news, and I see some people with masks, and I'll even end with one piece of result which will come as a personal communication. In 1960, the group of Hallberg, they were looking at gram-negative infections, and they gave mice E. coli endotoxin, lipopolysaccharides, to see what happens with regards to the immune response over light and darkness. And they got some fantastic, striking results. When they looked in the afternoon around 5 o'clock, almost all the mice died. You give the mice LPS at 5 o'clock, you kill them. You give the mice LPS at midnight, you have to come at night, but they all live, almost, all of them. Amazing. So we have a whole project. I don't have time to talk about it, but I will show one piece of preliminary result which may explain. We repeated these experiments of Hallberg. When we gave the LPS in the afternoon at 4, like them, and we waited 20 hours, we look in the blood, there's a huge wave of neutrophils and of monocytes. When we gave at midnight, no wave, even a reduction. And we want to know the reasons, and I'll give you a flavor of what we found. Now, I spoke about asparagus. I spoke about mice. What about people? Here, you see circadian rhythms are important for host immunity among people as well, surprisingly. And what they did here, they gave BCG, which is given to infants to fight tuberculosis. I got it when I was young. There was the Mantoux test. I was 0. I got it. But here they gave it to adults in Holland, to quite a few of them. I think a few hundred people, 302 volunteers. And they gave it either at 8 o'clock in the morning, early bird, noon, or evening. And they have very different results. The levels of IL-1, beta, interferon are much higher, and what is called trained immunity is much more effective, not only morning compared to night, also morning versus noon makes a difference. And I will tell you what we think is the reason. In mice in the afternoon, the levels of the steroid corticosterone peak, and we think this is the reason for the high mortality, while at midnight, melatonin levels peak, and melatonin is anti-inflammatory. While in people, the relevant steroid, which is called cortisol, peaks at 4 or 5 a.m. in the morning, and there's a school of thought that higher levels of heart attacks and strokes happen early in the morning for this reason. Therefore, now comes the personal communication. My wife and I went for the COVID-19 vaccine at 8 o'clock in the morning, all 5 times now. And while n equals 2, we were both not sick, while all our 3 kids and half my lab was sick. So early bird has advantage, and there's no risks involved. You only have to wake up early in the morning, and this is something I recommend, and this is one of the take-home messages from this talk. So to summarize this part of the talk about light and darkness, while we usually operate either during the day or during the night, this is not the case for our Earth that has a split personality because it's half dark and half in daylight. But unfortunately, with modern time, we have a lot of light contamination, which is not good for us because melatonin is not synthesized when there is light, only in the dark. And our modern way of living with a lot of electricity at night might be a hazard as well. Now, coming to the time and sorting. In science, we're only interested, like in court, in the truth, the whole truth, and nothing but the truth. And we want to know what stands the ultimate test of time. And what did we publish that is still true and relevant? So when I joined John's lab, this is the model that we developed. And I say we developed, but there was already a preliminary work before I came done by Suzanne Camille Reed together with John. But they had only 0.1% engraftment in SCID mice, and the cells were not known, except for a few myeloid colonies. That was it. And attempts to increase the level of repopulation and the fidelity and the reproducibility were not successful. Still, I prefer to go to John because the model he established is based on conventional bone marrow transplantation. And I thought that's the way to go. This is a functional model, and it's easier because the model that Irv developed has its advantages with human microenvironment and the thymus and fetal liver, et cetera. But here it's much easier. You transplant the cells IV into the circulation, and they have to function. So it took me some time and some tinkering to first irradiate the mice and wait one day to have less residual host immunity, to increase the cell dose. Later on, the cytokines to give them by IP injection daily, Saturday and Sunday for an entire year. But as you will see, from 0.1%, we got something very impressive. In '92, 30 years ago, this is the anniversary. This is my first paper with John. And this is what we claimed, that we have immature human cells repopulating the mouse bone marrow with differentiated human cells of multiple myeloid and lymphoid lineages. We looked at CD19 pre-B cells because that's where they developed in the bone marrow. And from 0.1%, we got mice with over 80 or 90% human cells, which was a big deal at the time. And here are words of wisdom coming from John. These systems allows the detection of immature human cells, identification of the growth factors that regulate them, and the establishment of animal models of immune hematopoietic diseases. We have our prophet from Toronto. And you can see it. You can see the CD19 pre-B cells. We now know why they're such high levels, because the levels of SDF1, the chemokine CXCL12 in the mouse bone marrow, are much higher than in human bone marrow. Here you see historic BFUE, a human BFUE, where the precursor comes from the mouse bone marrow, a mixed colony, CFUGM, there's some megakaryocytes here, there's some macrophages. This is all original, the first. Now, already in '92, I asked myself, how do the stem cells find their way home? I was about to go back to Israel, and I was facing a similar challenge to the stem cells, and I wanted to know, especially that this is with the mouse microenvironment and human stem cells, so this is something I was thinking a lot about. And it's not a coincidence, because the best example for homing comes here from Canada, when the Canadian salmon comes from the ocean back to the river of its birth to reproduce. So like it's a stem cell migration and self-renewal, and they give the next generation. The only caveat, they have to be careful not to become lunch for the bears who do the natural selection and take all the old and weak ones. And with all respect to the marathons that Peggy showed you, they have to go against the current to jump, and this is something of the eye to see. Now, we know for the immune system that immune cells respond to signaling molecules which are called chemokines, and that's how they find their way. But still, we didn't know how do stem cells find their way. And we talk about transplantation and the model that is based on transplantation, but we know from the elegant work of Anna that the stem cells, not only during transplantation, which is a clinical process, during development, they migrate, they home from the fetal liver to the bone marrow, and they're motile cells that can home. And this is the reason why all the hematopoietic stem cell work that we're talking about, and in the clinic, there's only hematopoietic stem cells which are clinically transplanted. You just throw them into the circulation, and they find their way home. No other stem cell knows how to do it. Unique. So here you see cross-section of the human bone marrow, and you see the bone-forming osteoblasts. And again, David has some seminal and important work. And all the brown that you see also around the blood vessels, this is this chemokine, which attracts, we think, attract the stem cells to the marrow. So in 1999, and this is after I returned to the Weizmann Institute, I stayed with John until '94. And I had some difficulties in publishing this work, and John was very helpful, and I'm very grateful for that. And we not only found how they home, we also found how they mobilized, and the involvement of adhesion molecules, et cetera. And now we have a full model, because we can do serial transplantation and monitor self-renewal. We can upregulate CXCR4 by 1 to 2 days cytokine stimulation with KIT ligand or with IL-6. And I was extremely honored and pleased that after I trained with John and he mentored me for 4 years, he asked for my help to do serial transplantation in one of his works. And he reproduced our results. So his kosher stamp means the world for me. And with this now, the model, there are many other discoveries that we made, which I don't have the time to talk about. I'll only say we found dopamine receptors on hematopoietic stem cells, human, were the more primitive cells, higher levels, and receptors for epinephrine and norepinephrine, and the crosstalk between, et cetera. And activated myeloid cells and lymphoid cells can secrete norepinephrine and dopamine. So we always say neuronal and hematopoietic, but hematopoietic stem cells also respond to melatonin, as I've shown you. So the boundaries are not clear-cut. Now comes the next challenge. After the work with the normal cells, we wanted a model for AML. Again, before I came to John's lab, Christian Sirard could engraft human AML cell lines, but not primary cells, did not work. Again, we tinkered with the model. We had a lot of technical problems with frozen cells. They didn't thaw properly, et cetera. Luckily, we had a very devoted collaborator in Montreal, Trang Wang. She finally sent us cells by bus. I took John's bicycle to the bus station. I took the cells, I transplanted, and the rest is history. And what you see here is in the mouse bone marrow. Human AML blasts, and there's one neutrophil that is squeezed. There's another neutrophil, actually, but it was left outside the paper. Here, there's an Alrod. Here is eosinophilia with inversion of chromosome 16. And more importantly, the more aggressive AMLs, M4, M5, which infiltrate the liver, we recapitulate that in the liver of the mice. We see blasts in the circulation. We have a functional model for human AML, which I think is the biggest achievement of my entire scientific career, just after 4 years with John. And here's what we claimed. And now we revisit 28 years later and see if what we said is true or not. We fractionated human AML cells on the basis of cell surface marker expression and found that the leukemia-initiating cells that could engraft SCID mice to produce Lambert colony-forming progenitors were CD34 positive, 38 minus, while 38 plus cells and the 34 negative fraction contained no cells with this property in some patients. We knew already then it's not in all the patients. And in order to sort the cells, there was no cell sorter in Toronto at the time. We had to go to Roswell Park in Buffalo. And for that, you need to cross the border to the US. At those times, if you were a Canadian, John shows his driver's license, he passed. I had to fill up forms with an Israeli passport and stand in line with all the criminals and suspects. And John said, when there was the 50th anniversary of Ash, he said the American Border Patrol had problems with Tsvee's Hebrew passport. It was not with me that they had the problems, it was the people ahead of us. And we went, we sorted, we had chicken wings, we went back, and I was in the way. And again, words of wisdom from John. This in vivo model replicates many aspects of human AML and defines a new leukemia-initiating cell, which is less mature than human colony cell. And I like this definition much more than cancer stem cells that nobody knows exactly what they are, but there are the cells that initiate the disease, and these are the cells that one would like to eradicate. And John told me at the time that it's not only the first with the human leukemia or with any human malignancy. He told me it's with any malignancy. In mice, in rabbits, in rats, this is the very first. So it's very rewarding for me, and John told about me at the time that I was the right person at the right time at the right place. I say the same things about him and his lab. Now, we want to know more about the model, and we have shown that CXCR4 and CXCL12 are also regulating this model, this functional preclinical model. And in parallel to us, John has shown that CD44, which is an adhesion molecule downstream to CXCR4, just like VLA4, also is essential. And we have a paper about CD44 with regards to SD1 with normal stem cells. So while CXCR4 and CD44 are essential also for normal stem cells, the malignant cells are more dependent on these interactions for a wide variety of reasons. The malignant cells, they also destroy the microenvironment. David has an elegant paper on that, and Dominique and Christina. And so what we could do is we could selectively purge the malignant cells while maintaining the normal cells there. And I think John the same, and I still hope that it's having clinical relevance. Now, I told you the niche protects stem cells from chemotherapy because stem cells on their own, they're not protected. Only when they interact via adhesion interactions. And one of the important molecules here is again CXCR4 and SD1. The group of Nagasawa have shown that they're both essential for cell cycle and for quiescence and for chemotherapy resistance. You don't have CXCR4 in the cell will cycle forever, et cetera, and you need the SD1 for the adhesion. And another molecule which is involved is EPCR, which was originally identified by Ihor Lamishka in fetal liver, and people use it simply to enrich the stem cells. But EPCR signals via PAR1, anti-inflammatory, anticoagulant. It's APC, EPCR PAR1. And together with Chuck Esmond and Wolfram Ruff, who are experts in the field, you can see that if we neutralize EPCR, the mice are not chemotherapy resistant. They die within 2 weeks, while with a non-neutralizing antibody, they will all live. EPCR knockout is lethal, but there is a mutation where they have low levels of the gene, Procer low. You can see they cannot compete for long-term repopulation. And the reason why I'm showing these results is if you look at human KG1A, it's an AML cell line which was used by Kurt Tsvee to identify CD34. Look at the huge amounts of EPCR, not only on the primitive cells, but also on the committed progenitor cells. And unfortunately, patients who have the normal stem cell surface markers, they have low responses, and it's a poor diagnosis factor. And PAR1, which is downstream to EPCR and also to CXCR4, is also important for leukemia. And CXCR4 expression is a poor prognosis marker in human AML. And the main prognostic factor for complete remission in AML patients was lower CXCR4 expression. Overall survival was negatively influenced by higher CXCR4 and downstream VLA4 expression. And our biggest hope, both John's and I, that this preclinical functional model, which already provides a lot of information on what happens in the clinic with transplantation with the normal cells, or with regards to malignancies, will help to develop a cure. And we're living in a time where things move much faster, and we are still hopeful and optimistic. Now, this is a historic picture from 1992, when John's hairstyle was more blondie oriented. Even I had something here. And this is Monica and Francoise, which are also played a major role in the normal hematopoietic stem cell work. This is Joseph Vormor, who is an MD, and he played an important role in the leukemia work. And I would like to congratulate John again, and also Stu, and to say that for me, to speak or to be introduced by John, first time it happened in my life, I choked. I couldn't say a word. And how grateful I am for his friendship, for his mentorship. All my career started here with these two seminal papers that did so many tricks for me. So finally, I'd like to thank all our lab members and all our collaborators. Thank you for your attention. Shalom.
Man, it's lovely to see that walk through. You know, we don't often see the founding story that led to, as I say, us standing on this platform. So it's lovely to see that and to see some of these old pictures. So thank you very much for the walk through, Tsvee. It was a real terrific to see that, and you're the perfect person to have told that story. So any last question that we have? We won't go to the question. We'll end with Kristen. So thank you very much, Tsvee.
Thank you.
Hi, everyone. I'm Dr. Kristen Hope. I'm a senior scientist here at the University Health Network. I'm an associate professor at the University of Toronto and a medicine by design investigator, just here to deliver some closing remarks. I know I'm between you and lunch, so I'll try and be pretty quick. I just first want to congratulate John again on this fantastic award. Very, very well deserved. I was fortunate enough to train with John for my PhD, and it was, I can echo Dr. Lapidot's words in saying that it was an absolutely foundational experience for me as a scientist, and has set up my career as well. So thank you, John. I want to thank today's chairs, Drs. Jim Woodgett and Dr. John Dick, of course, And I really want to thank all of the incredible speakers who have traveled really across the world to be here with us today and to share their really important work. I'm sure you'll all agree with me that this was an absolutely fantastic symposium that I know I enjoyed very much. So thank you again. Thank you to the Gairdner Foundation for, of course, hosting this event as part of Gairdner Science Week, and to all of the Gairdner Science Week partners and sponsors, without whom this event, of course, wouldn't have been possible. The Gairdner Foundation would love to hear from you. Please kindly fill out the survey at the end of the digital program, which you can go to using the QR code that's on the signage and screens that you see around this week. Gairdner Science Week continues with a lineup of really fascinating events over the next couple of days. So to access that schedule, go to gairdner.org, or you can follow along on social media at Gairdner Awards for event schedules, details, and live coverage. Thank you.